Okay, let us just rewind a little bit. We are looking at how the reactive species could play a role in the signaling processes in the cell. And so that is different from the negative aspects that we've been seeing so far about reactive species, which are much better known. Essentially, they act by modifying the proteins. Right? They act very fast. They are very reactive. So they modify the proteins and the processes, the various cyclic processes in the cell, make it possible for signaling to happen. We are going to see some examples of it. Essentially, the changes that happen, the chemical changes that happen because of the reactive species, in other words, oxidation process. Major examples, because this is what is known most about, are changes to the iron sulfur centers. Fe can be oxidized or reduced by reactive species. So the proteins that have iron sulfur centers can get affected. Thiol groups, as in cysteine, they can be oxidized or reduced either singly or in pairs by reactive species. Histidine groups, they can be oxidized to form oxohistidine. So these are the major things that are known. So we will talk about these. I don't rule out the other possibilities. Only thing is that they are not very common, or at least they have not been studied extensively. And as we saw in the last class, oxidation and reduction of the proteins could influence their function either directly or through a transcription factor and so on and so forth. The indirect means could be quite a few. Through transcription factor by mediating its effect on another protein which is dependent on this protein and so on and so forth. Okay, we'll see some examples. So the transcription factor and thus the production, which is transcription directly of the relevant proteins such as OXR, OxyR, and so on and so forth. Okay. So we saw this again in the last class, the molecular level action of oxidation reduction of iron sulfur centers, thiol groups or histidine has an effect on at the cell level or the pathway level. And we are first going to see some examples in bacteria and then in mammalian cells. The first example which I have already seen is the oxidation of iron sulfur centers by superoxide, for example. Okay. SOX R is the one that has the iron sulfur centers, so it can sense in a way in a manner of speaking, it can sense, let us say, superoxide in the cell, something that can oxidize it. So SOX R protein activates a transcription activates transcription of SOX S gene to produce a SOX S protein, and the SOX S proteins activates a production of about 100 different proteins that respond to increased reactive species levels, especially the superoxide levels. For example, superoxide dismutases, DNA repair enzymes, and so on and so forth. So this is the way the signaling gets implemented through reactive species. One example. And this is the figure from Diotro and Toledano. The inactive form, I would have preferred the protein to be separate from the DNA. This is the figure from their picture. In my book, I have the protein separate from the DNA. So you have the SOX R, which is a 2Fe2S. This is a plus thing, right? This is just plus, right? So if it is plus, it doesn't bind to DNA, it is inactive, and therefore, rather, it doesn't bind to DNA, and therefore it is inactive. When you oxidize it with superoxide, this becomes 2Fe2S2 plus, right? Loss of electrons is oxidation. Superoxide pulls out an electron from this and it oxidizes. When it oxidizes, its conformation changes and that helps it bind to the DNA and become transcriptionally active. So that's the way it works. And you should also appreciate that oxidation causes this. And the reduction of this protein through a series of electron transfers which are mediated by an ADPH dependent paradoxin, you get back the inactive form. So you have a cycle that is set up which can be seen in a signaling context, right? So you get a general picture. Oxidized SOX, this is the reverse part. Oxidized SOX R is reduced by an ADPH through a series of electron transfer reactions. Yeah.
Since DNA is already negatively charged, won't one positive charge socks are be enough to interact?
Charge wise, yes, but interaction depends on so many different things. The DNA is supercoiled, right? So while binding, there is a certain rearrangement and things like that, and then it binds. So a lot of the matching of, pardon, yeah, the conformation is more important. In the conformation, yes, what you say, the charge is important, but whether the DNA is able to see this charge is going to be inside. It's going to have, it's a cluster inside the protein. Whether that is going to be effective as an overall molecule and things like that. Yeah, it certainly it has a plus charge at the center. Whether that is going to be effective and things like that need to be looked at. I don't know the details. It doesn't happen is something that I can tell you for sure. And I also know that conformation is a lot more important than this. The other things, are guesses, so you can fill in the gaps. Whatever the molecular details, fill in the gaps. I'm not a molecular biologist. Yeah, and we kind of go and make pictures and things like that. Coming up soon is a picture that we made which is completely wrong, and so on, which was a little surprising to Jayashree, right? How can it be wrong? You're wrong a lot of times. The only thing is that you should have the humility to say you're wrong, and which is completely acceptable in science. It is based on certain principles. Any other questions, comments? Okay. Second example, oxidation of thiol SH group in the cysteine 199 position in the oxy-R protein. Oxy-R protein happens to be a transcription factor by, let us say, hydrogen peroxide, leads to an intramolecular, inside the molecule, disulfide bond formation between 199 and 208. So you become, your S forms another S, rather forms a bond with another S. So you get an S dash S between cis 199 and cis 208. This leads to a conformation change of oxy-R, which promotes its binding to the DNA to promote transcription. Okay, so that's the way it works. And from the paper, oxy-R represented, the thiol groups are represented here. This is gone missing. It's come back. Thiol groups are represented here. If it is in this form, then it is inactive. Oxy-R in this form is inactive. For example, hydrogen peroxide, not just hydrogen peroxide, but for example, hydrogen peroxide can convert that into a disulfide bond. When a disulfide bond forms, it becomes active, it binds to DNA and promotes transcription. It's a transcription factor, transcription of other proteins. And to get this back, the inactive form, you need to replenish the pool of inactive forms after the work is done. The reverse happens in the same, pretty much the same way as the earlier example. Glutaredoxin 1, that's GRX. Right. Okay, third example, oxidation of cis zinc redox center in HSP, heat shock protein 33, by, let's say, hydrogen peroxide plus heat. This is what the data is known about. This works for the heat shock protein 33. H2O2 plus heat or HOCl. Okay, this is what is causing the oxidation, let us say. This causes formation of two disulfide bonds, and the zinc gets released. The zinc is in the center, that gets released because of the formation of two disulfide bonds. And this process activates the heat shock protein 33 chaperone. This is a representation, right, of the process. The HSP33 monomer is an inactive chaperone. Heat plus H2O2 or HCl converts it into a dimer with two disulfide bonds and so on and so forth. I mean, rather when it has this, then it forms a dimer, and the dimer is an active chaperone. Yeah, this could happen in many different situations. So, as an example, this is given. I think there's one more related example in bacteria. We're still in bacteria. Diamide mediated oxidation of cis zinc in RSRA protein. It's an anti sigma factor, RSRA. This leads to formation of two disulfide bonds along with zinc release, similar to the earlier case, which results in a loss of a sigma R binding by RSRA, right? And this is what results. You have the RSRA sigma R complex, and this, in the presence of diamide, releases zinc, and you have formation of these from this. This releases the RSRA from sigma R, and that leads to the activation, because when this is bound, it is unable to bind to the DNA and cause transcription, whereas when this is released, the sigma R can bind to the DNA and cause transcription. Okay. So, slight differences in themes, that's all. The principle is just the same. Reaction, oxidation, conformational change, and whatever happens as a part of that. And of course, it's usually a cyclic thing, so you get back the original reduced form by some mechanism.
What is an anti-sigma?
Okay. I'll tell you what I know. Okay. We have worked with sigma B and so on and so forth in the context of shear effects that gets mediated through sigma B. It's a transcription factor. Sigma B is a transcription factor. Uh, Sigma R seems to be a transcription factor going by this figure, right? It binds and it causes transcription of other things and so on and so forth. Say RSRA, sigma R complex, sigma R DNA binding causes the activation here. Here, RSA, sigma R complex, sigma R DNA binding is not possible here, and therefore it causes inhibition. Okay. What exactly sigma R does, I don't know. Rather, it's known. I'm sure it's known. I just don't know. I have some more experience with sigma B because we have worked with it and so on and so forth. I have not read about sigma R. So please read about sigma R. If you find something interesting, please share it with us. If it is in the context of reactive species and all that, we'll all get to know. And the next time somebody asks me this question, I'll have something to talk about at least. That's right. We did extensive studies with sigma B. That's why I always collaborate with molecular biologists, molecular people. It is a cell biology, molecular biology kind of a thing. So they provide the mechanistic angle to our work. I look at it from an engineering point of view. I'm basically an engineer, so I want to manipulate, but at the same time, I want to understand what happens. So we had this was the work with Susmita between 1999 and 2004. 2000, she graduated in 2005. No, 2004 itself, I think. So yeah, she was bright. She was bright. She's now a professor at Mount Sinai Hospital and so on and so forth. I mean, the programs associated with the Mount Sinai Hospital. Very bright person, and she did work on shear effects on bacteria. So I went to KK Rao in Biotech Center and said, "Can you help us with this and understand this process?" We have, I mean, he was a co-guide right from the beginning because I wanted to understand the mechanistic details. So he had uh, directed us, or he had led the studies on sigma B related thing, because sigma B is a known factor, known transcription factor, stress related transcription factor. So he said, "Why don't we look at sigma B?" And then he figured out how to. He already knew that he was already working with sigma B. By the way, he is Madhulika's guide, KK Rao. So next building, so just walk across, and we had set this collaboration up, and we are authors on all the relevant papers. So Smita wrote, I think, 5 papers, very good papers. And both of us are authors on them. So that's the way it works. And if you want to make significant enough contributions, collaborations become necessary, because you will have to be at cutting edge of everything if you want to make a decent enough contribution. So that is rather difficult to be at the top of everything. If you do research alone, maybe, but even then you'll get bored. Here we do teaching, research, administration, and so many other things, and so on. So that becomes a little, and we'll have to have our own lives. So that becomes a little difficult. And I have, my passion is more of the learning process. I don't call it teaching. More of a learning process, and so that's why I never get bored of courses. I run courses for many years, never get bored, because each batch is different. So that way, I mean, I'm excited to experience different things with different batches. Always, I'm always worried about that part of the thing and whether things are getting across and so on and so forth. I have a means of figuring that out. Not everybody does. So, any other questions, comments? Good. Okay, so that's, I think this is the last example. The oxidation of perar. Perar is another protein which Surabhi worked on, one of my older students at IIT Bombay. It happens to be a functional ortholog of oxy-R, which means that an ortholog from a gene deviation point of view and so on and so forth. They have similar functions and things like that. You can look up what an ortholog is. Oxidation of perar by hydroxyl, let us say from H2O2 or something like that, releases iron and leads to the loss of binding between perar and DNA. Perar is again a transcription factor, perar and DNA. So you have, when this is bound, when perar is bound, there is a repression, there is no transcription. And when the oxidation happens, the perar gets released because the histidine becomes oxohistidine here. The oxidation happens. When the oxidation happens, the perar gets released, and then there is derepression, which means the protein gets, it's just the opposite of transcription factor being an activator. Here, when there is a release, transcription happens, and that's what happens. Okay. Okay, so some examples in bacterial systems, not exhaustive, but the most common ones.